connective tissue forms mucous membranes. Because a mucous membrane is a layer of epithelium and a layer of connective tissue. What type of connective tissue? It's always the same type of connective tissue under any epithelium in the body. Areolar. What's the name of that areolar connective tissue forming our mucous membranes? It was the one thing I wanted you to learn from 430, if nothing else. And why I chose that, I don't know. But I know I asked you that question on quizzes again and again and again. And you forgot it. LP. Somebody, please. We went over it so many times. Lamina. Lamina propria.
That's true.
That winter break was just a little too long. All right, so there's your quick recap discussion of difference between type and name. We didn't talk about synthesis and storage of thyroid hormone when we covered the classes of hormones earlier. We said we would discuss it later. Now is later. And the steps are shown on these diagrams. Slide 61 shows the steps. Slide 62 shows the steps. They're in the study guide. So you've got thyroglobulin. If you see the term globulin, what does that tell you? It's a globular protein. It's a protein. Thyro, because it's in the thyroid. So thyroglobulin, this huge thyroid protein, is synthesized in the follicle cells and then shipped into the colloid. And how would you ship a big protein from the inside of the follicle cell across the membrane into the colloid? Exocytosis. Later, when it's time to bring in that same large protein, how would you do that? Endocytosis. So we move that protein into the colloid. Iodide, not iodine, if we want to be really technical here, iodide is what is taken up by the thyroid tissue. Gets moved into the follicle cells, moves through the follicle cell, and then crosses the other side of the membrane to enter the colloid. So technically pumped across the basolateral membrane, that sets up a concentration gradient so that it can just then diffuse across the luminal membrane. And remember, the luminal membrane faces the lumen, right? The open space within some hollow structure, like the inside of the follicle. So pumped across basolateral, moves through the cell, and then across the luminal membrane, joins up with that big protein, thyroglobulin, where we already have lots of tyrosine. And remember, that's what we make the amines with, or at least catecholamines and thyroid hormone, tyrosine. Two molecules, two tyrosines joined together. The iodide is oxidized into iodine, and then either 3 or 4 iodines get attached eventually to the two tyrosines that are coupled or joined together. So if you're curious what MIT and DIT are, this is one tyrosine with one iodine attached. This is one tyrosine with 2 iodine attached. Diiodotyrosine, monoiodotyrosine. So then if you take one tyrosine that has 2 iodines, join it with another tyrosine that has 1, what have you just made? T3. Thyroid hormone, the version called T3, or tetraiodothyronine. Okay. What if you join 2 DITs together? Right, 2 tyrosines that already have 2 iodines attached. Now you just made T4, tetraiodothyronine. So that's how we make thyroid hormone. We take the tyrosine that has 1 or 2 iodines attached, join it to another tyrosine that has 1 or 2, and then you come up with T3s and T4s.
You said T3 was tetraiodothyronine, or iodine. You said tetra for T3, and then you said it for 4.
Of course I did.
So
T3 is tri-3 thyrine, and T4 is tetraiodothyronine. I did get more sleep last night, but still keep an eye on it. It's always a good idea for this first class. Once inside the follicle cells, we can take up thyroglobulin with all of these T3s and T4s attached to it. We take it up through endocytosis. But once we do that, T3s and T4s are cleaved from thyroglobulin. Think of it as like a big docking protein. Right? It's just a way to temporarily house lots of molecules of T3 and T4. But it's housed in the colloid. We have to get it into the cell, endocytosis. Then we use enzymes to break the T3s and T4s off of thyroglobulin. And now they are free to diffuse by simple diffusion, right? And that's what we have assumed for quite a while now. Like I said, new research shows maybe there's transporters involved, but then can diffuse into the blood. And once it gets into the blood, can it dissolve in the plasma? So what's going to happen?
Another transport.
It's going to have to bind to a carrier. Albumin is a carrier. Albumin does many things. That's not its only function, but it can carry thyroid hormone. There's another one called transthyretin. It transports thyroid hormone, transthyretin. It's another plasma protein. TBG, it's binding globulin. I think it's thyroid binding globulin. If I don't have it on there, that's what we'll go with. Or you could think thyroid hormone binding globulin, that would work too. Mostly what's released is T4, not T3 as you would expect, because T3 is much more biologically active. So what ends up happening when all this T4 reaches the target tissue? It gets converted to T3 before it actually binds to receptors at the target cell. So all you need is an enzyme, a deiodinase, to basically remove one of those iodines. And you very quickly converted T4 to T3. That typically happens at the target tissue. Hormone actions. Well, first, stimulus. What is signaling follicle cells to secrete more thyroid hormone? TSH. Well, what's causing TSH to be secreted? TRH. Okay, so you have to go to the beginning of the pathway. What's causing TRH to be secreted?
No TH.
There really is no stimulus, not in adults. And that is why thyroid hormone levels tend to be very, very stable. They don't tend to fluctuate. Now, in infants, cold is actually a stimulus. And in turn, when thyroid hormone levels go up, what happens to heat production? It goes up. And that's because thyroid hormone controls metabolic rate. Right? It basically sets your BMR, basal metabolic rate. So why, when adults are cold, don't thyroid hormone levels go up, but in infants they do? What do we have that infants don't? We have something that can generate a lot of heat very quickly, a lot of skeletal muscle mass. And because infants lack that skeletal muscle mass, they have to have an alternative means of generating heat. So they have to rely on whole body metabolism. We can generate heat much faster just by contracting skeletal muscle.
Wasn't there like a certain cell that's sort of like a fat in infants?
Brown?
Yeah.
Brown fat?
Yeah.
and I forget how to describe it exactly, but ATP synthesis is uncoupled from, I can't remember what it is, but basically you are you're turning over ATP simply to generate heat. You're not actually using the ATP to do cellular work. So just burning through fuel, making ATP for the sake of generating heat. So infants have brown fat. And thyroid hormone would certainly increase the rate of ATP turnover in that brown fat. So yeah, thanks for bringing that up. That's something infants have that we don't. We don't need it. It's not there to store nutrients. It's not there to store energy. It's actually there to produce fat, to produce heat. How about a break? 9:25.